Welcome to this lecture on the immune system. We will specifically be looking at the adaptive immune response of the immune system in this lecture. I've posted a video about the innate immune response to D2L that you can watch and take notes on, but this lecture is really going to focus on really what we think of as the true immune system, is how we gain immunity from viruses and bacteria and other pathogens. In this lecture specifically, we're going to first start off by talking about how our body distinguishes self from non-self. In other words, how does the body know which cells belong to us and which cells are foreign? We'll also talk about the fact that the adaptive immune system can respond to pathogens in two different ways. This is the antibody-mediated response and the cell-mediated response. And as we discuss these types of responses, we'll really be looking at how the adaptive immune system works. And we'll wrap up lecture by talking about what is immunity? What does it mean to be immune from a pathogen or disease? So first off, it's really important that our body and our immune system be able to determine which cells belong to us and which cells are considered foreign invaders. And the way that we do that is through markers that exist on the surface of our cells. And major histocompatibility complex marker. Histo means tissue. So really, our body is looking for these markers that tell us that our tissues are compatible with ourselves. These MHC markers, as kind of highlighted in this diagram, are present on the surface of every single one of our body's cells. And this is how we tell our immune system, hey, this cell belongs to me. What's really cool about these MHC markers is they're different from person to person. So unless you are an identical twin, you have different MHC markers from your brother, sister, parents, aunts, uncles. So every single person has different MHC markers. This is why things like tissue transplants can be really tricky. This is why organs, for example, can be rejected when you have an organ transplant, is because you have to find MHC markers that match, that won't be rejected by your immune system. So this is how our body determines self, by using these MHC markers. Foreign invaders, they have a different type of molecule or protein or substance on their surface that we refer to as antigens. Typically these are proteins, but it can be carbohydrates, it can be substances that are secreted by those pathogens, but basically these substances are not recognized by our immune system. And so when our immune system sees these, it's going to trigger an immune response. Right now, for example, people are receiving a vaccine for COVID-19. And that vaccine is actually asking your cells to make an antigen. So your cells are making a protein that's normally found on the surface of the COVID virus. So it has these kind of little spike proteins that make it look like this virus has kind of a crown or corona. Your body is going to make these spike proteins. Those spike proteins are going to be recognized as an antigen by your immune system, and they're going to mount an immune response. And we'll come back to how that gives you immunity here in a little bit. So MHC markers allow us to determine when something belongs to us. Antigens allow us to determine non-self cells, cells that should not be in our body and need to be destroyed. So the adaptive immune response, this response really happens when the innate immune response fails. And again, this is the YouTube video I posted on D2L. And you can read or watch more about the innate immune response and take notes by watching that video. But when that system or when that response fails, that is when the adaptive immune response kicks in. And it's really important to remember that this response is specific. So it's going to specifically attack whatever pathogen is infecting you. And secondly, really importantly, and something that the innate immune response does not do, is the adaptive immune response, once it's done fighting that pathogen, it also provides memory. So it prevents that pathogen from attacking again. It is possible that the pathogen can mutate or evolve. Sorry, I misspelled evolve there. And in that case, if the pathogen is different enough, our body, when it sees that new mutated version of the pathogen, it will need to mount another immune response. It won't recognize as having seen it before. This is partly why we have to get a new flu shot every year, because the flu virus literally evolves very quickly. But again, kind of going back to the big picture of our immune response, remember that our innate defenses are really our first line of defense. We have those physical and chemical barriers on the surface of our body. If the pathogen penetrates those barriers, we'll have internal cellular and chemical defenses happening, our second line of defense. But if this pathogen survives both of those, that's when we're going to finally start our third line of defense, which is our adaptive acquired immune response. So before we get into what's actually happening here, it's really important that we actually are aware of the types of cells that function in this response. So lymphocytes, these are cells that are going to perform kind of the majority of the work that's required for the adaptive immune response. Cyte means cell, and so lymphocyte is really just telling you that these are cells of the lymph. We'll talk more about our lymphatic system later in the semester, but these are cells that are going to be found not in our circulatory system, but in our lymphatic system. And there's two types of lymphocytes that we are going to talk about in this lecture, and that's the B lymphocytes, or you can just shorten that to B cells, and the T lymphocytes, which are it's often just shortened to T cells. Both of these types are specific to the antigen they recognize. So whether you're going to have these cells here, often call these receptors, but these receptors are going to be very specific. So one cell, for instance, might have receptors that are triangular in shape. Maybe it has receptors that they look different. So we have different receptors. So those receptors are going to cause these lymphocytes to be very specific to the antigen that they structures here that are responsible for recognizing antigens. The terms B and T, those terms help to tell you where these cells mature in the body. I'm not going to go over that because I just think we don't need to provide that much detail, but just think there are B and T cells. Both types have these receptors on the surface of their cell that are going to be responsible in recognizing antigens. And those receptors are very specific. So we have different receptors for different antigens. There are basically two things that are going to happen when either of these cell types sees their antigen. For one cell. So I'm going to erase my picture here really quick so that I can draw another. So let's imagine that we have our B cell here, and it has draw kind of a red triangular receptor to an antigen that's sitting on a bacteria, and we'll make it look like it's a mean bacteria, right? Because it's trying to make us sick. So it's got these triangular shaped antigens on its surface, and our B cell recognizes that we have a foreign invader. So what's going to happen here is that this cell is going to divide into two populations. It'll divide into effector cells and memory cells. The effector cells, you can think they cause an effect in the body, and specifically those cells, bacteria that they can find, and they're going to destroy those bacteria. So those really kind of mount a defense against these invading pathogens. The memory cells, they basically hang around in our body to remember the invader, so that if this guy ever comes back, they can mount a faster immune response. And that's really good because the faster you can mount an immune response, the less sick you're going to feel, because your body's not, it's going to be really easy to just kind of nip it in the bud and get rid of it right as you are getting sick. So we're going to talk a lot in this lecture about B cells and T cells. So just kind of keep these in mind. Again, they have receptors that recognize antigens. When they see an antigen, they're going to divide into two populations: the effectors, which actually go on and kill and attack any invading pathogen, and the memory cells, which will hang around for future attacks so that we can destroy that pathogen even faster and not get sick that second time. And really, our body is constantly scanning, or our immune system is constantly scanning our body for these foreign invaders. And there's another cell type that is responsible for performing the majority of the scanning, and that is the macrophages. The macrophages is kind of shown in this funny comic here, which if you want some funny anatomy and physiology comics, check out the Awkward Yeti. They also have an Instagram, it's hilarious. But basically, the macrophages are constantly scanning our system, our entire body, and if they find an invader, they literally eat it, as is shown in this comic, and they then alert what are called helper T cells. These helper T cells are going to then start an immune response. And there's two types of responses that we can have. So the first type of immune response that we can get is, and this is specifically going to be carried out by B cells. So we have kind of this game of telephone. We have our macrophage that's now told a helper T cell that has now told a B cell. And we have kind of another branch here that we can talk about here in a moment. But in an antibody-mediated response, it's the B cell that's now going to react. And antibodies and mount immune responses against antigens that are freely floating in our body fluids. So these are things that can be floating like in our lymph, in the liquid of our tissues, in our blood, in our mucous membranes. So these are going to be things that are physically have entered our body and are just kind of floating around in our fluids. And the way that B cells work is, again, let me change my color here, which I'll draw. And remember, different types of B cells, but each B cell has those, what we were just calling receptors on the surface. The actual name of those receptors, to add a little more detail, those are called antibodies. And it's the antibodies that are responsible for recognizing foreign antigens. And there's lots of different antibodies. In general, an antibody is kind of this Y-shaped protein. So if we were to kind of zoom in on the structure of an antibody, they make kind of this Y-shaped protein. But we can have different types. Sometimes it could be, say, like circular, or we could have some antibodies, and it's the type of antibody that is responsible for detecting different antigens. So there exist. And so we have a lot of different options that our B cells can choose from when working to recognize and find these antigens. And again, different antibodies recognize different antigens. So for instance, are responsible for recognizing something that's probably triangular. Notice that triangle shape could fit in that Y shape. Circular antigens, and then say square-shaped antigens. So these would be We have 3 different antibodies that are Y, in general Y-shaped, and then they recognize differently shaped antigens. So that's the antibody-mediated response. So if the macrophage has found something floating through our body fluids, the helper T cell then goes on to activate our B cells, which say, hey, there's something floating around, can you please destroy that? The other pathway that we can get is the cell-mediated. With our macrophage, we're still activating our helper T cells. Oh, my pen is freezing, come on. There we go. We still have our helper T cells, but instead of doing the B cell route now, we're gonna do the next route, which is now we're going to activate those other T cells. And it's the T cells that mediate the cell-mediated response. This specific immune response protects us against cellular pathogens. So these could be things like a virus that have infected our cell, physically infected our cells, or it could be abnormal cells. So for instance, when our cells become cancerous, they start presenting different signals on the surface of themselves, which tells the body like, hey, I'm sick. We might need to consider getting rid of me. What's going to happen here is that the T cells go through a maturation process where they become what is called a cytotoxic T cell. And those T cells are then going to they are going to then attack infected cells and destroy those cells. So if a cell becomes infected by a virus or something has gone wrong with the cell, that is when this cell-mediated response will be activated in order to destroy that cell. And again, the Awkward Yeti just has amazing comics that really, they're so accurate and so funny. So check out the Awkward Yeti. So at this point now, we have seen that essentially what's going to happen is an antigen is going to enter our body. The macrophage is going to eat it up and it's going to say, hey, I need to get rid of you. And it's going to present basically its waste to the helper T cells. And those helper T cells can then go on to either start an antibody-mediated response or a cell-mediated response. So we've, in our previous slides, I've really talked about the threat, the detection, and the alert phase of the adaptive immune response. So just to kind of quickly review the first 3 steps, and then we're going to get into kind of what happens in step 4 through 7, the alarm, the building specific defenses, defense, and continued surveillance. So let's go through what I'd like to do to discuss the adaptive immune response is to actually physically go through this entire kind of diagram to see what's happening in more detail. This is a great place to pause if you are short on time and can't continue the lecture. We're about halfway through. So if you want to pause, go ahead and do that, or otherwise, let's move on. So like we've already talked about in our body has to determine that there's a threat. And this is specifically going to happen when an antigen or an organism without an MHC marker invades the innate immune response. So again, antigens can be anything from, say, the spike proteins that are on the surface of the COVID-19 virus, or they could be organisms, say like parasites, protozoa, fungi, prokaryotes like bacteria. So there's lots of things that can cause different disease states in our body. More commonly, like when we think of the common cold, we're being infected by a non-living virus. And then very rarely, for instance, if you've ever heard of mad cow's disease, these types of diseases are actually caused by misfunctioning very small proteins called prions. So it's actually it's a small protein that can wreak a lot of havoc if not destroyed in our body. So there's lots of different things that our body can recognize as being a threat. So again, once the threat has been detected, this is when macrophages are going to engulf that pathogen. This video actually shows a macrophage in gray engulfing a MRSA bacterium, which is green, and it's then going to go on to present the antigens on that bacterium. So again, really what's happening here, that macrophage is going to eat that antigen and the bacterium in this case that's connected to that antigen, and it's going to basically break that entire thing down. It's going to poop essentially all of that waste product out and present it to the helper T cell. So the helper T cell is literally looking at kind of the waste product of the macrophage and saying, oh crap, we have an issue, no pun intended, or I guess pun intended. And so at this point now, those helper T cells, which just like the previous cells we've talked about, they are specific to the type of antigen they recognize, that's now then where they can mount an alarm. Really, again, it's important to remember that there are thousands of different types of T cells that are helper T cells that exist in our body, because again, they have receptors on their surface which bind only to specific antigens. So because of that, the macrophage literally has to kind of wander throughout the body trying to find its correct helper T cell in order to mount an alarm. So this can take a bit. So being able to detect and basically chew up and then release and show these antigens to our immune system can take a bit. However, once this happens, that's when the alarm can be set off that we need to mount an immune response. So once the helper T cell has physically attached to the antigen that's being presented by the macrophage, that's when it then becomes kind of what we can call activated, and it's going to send the alarm to the B cells and the T cells. Basically what it does is it's releasing chemical signals that I'll just draw kind of as circles and little dots. And those chemical signals will diffuse throughout the body until the B cells and the T cells are activated. And this can take a bit. Steps 1 through 4, so everything from you have your threat, it's been detected, we present that threat, and then we mount an alarm, that can take 1 to 4 hours. So it's fast, but not super, super fast. But once these chemical messages have been released by the helper T cells, that's now where we can then decide what type of specific defense we need to react with. So at this point, this is where our body's going to figure out, okay, do we do an antibody-mediated or a cell-mediated response? But in both cases, what's going to happen is kind of what's being shown here in this diagram, is that as soon as the either B or T cell, in this diagram it could be either, as soon as they see that chemical, these are considered kind of naive or immature. And once they kind of have been activated, they're going to divide, as we can see here. And there's 2 populations that will be created that we'll talk about here in a little bit. But what's really important is that in this situation, when these cells divide, they have just created clones of that original naive cell. So now we've gone from having just one cell that can react to that specific antigen to now thousands of cells that can go on and find that invading pathogen and begin to mount an immune response. So that's really important. It allows us to basically kind of think about if you were going to, say, a bakery and you find this one type of cupcake that you love and you're like, okay, for my birthday, I want, from this display case, I want to choose this one cupcake, but I want 25 of those cupcakes for my birthday. And so the baker would have to take that one display cupcake and make now 25. This is essentially what our body is doing. It just has like a display case version of every single type of B cell. And then as soon as that specific type of B cell is activated, it divides and creates these clones. So now we have a huge, basically, amount of soldiers that can go on and attack that pathogen and mount an immune response. So again, because at this point, kind of our story splits where we can either go to antibody mediated or cell mediated, then our steps you'll see are divided into step A or step B. So we're going to look at what happens in the antibody mediated response first, more specifically, and then we'll look specifically at the cell mediated response. So if a B cell is activated, so if a threat and the detection and the alert has activated a helper T cell, which then goes on to activate the B cell, that's when we are going to mediate or begin the antibody-mediated response. And again, what's going to happen here is that once this helper T cell has activated or stimulated our B cell, so helper T cell is purple in this diagram, our B cell is blue. We can see its antibody on the surface here. Once this B cell has been activated, it's going to divide. And it's going to create two categories of cells from this division. Some of those cells are going to be plasma cells. Those are our effectors. So remember, we can have effector cells and memory cells. The plasma cells are going to be our effectors. Basically, what these plasma cells do is shown here, that they are going to actually secrete antibodies. So notice, up until now, those antibodies have been living on the surface of this cell. These plasma cells actually make a ton of antibodies and release them into the bloodstream. And that particular that particular antibody can then go on to bind to our antigen. So these are going to bind to the antigen. And basically, they kind of put our antigen in jail and kind of hold on to it until something can come and destroy it. So we're going to kind of talk about how that works here in a second. But essentially what we're doing is we've created these effector cells that are going to go on to eventually kill our pathogen. And this figure kind of down here, as well as this figure, is showing that process. So these kind of figures are one and the same together. If we kind of go back to that division phase of our B cell, again, half of those cells are going to become these plasma cells and the other half become memory cells. And we'll talk about those here in a moment. But what I want to do is actually look at how do these antibodies, if they're being released into the bloodstream, how do these antibodies actually work to kill our pathogen? Because that's the whole goal, right? We're trying to get rid of the thing that is infecting us. So the easiest way to remember how antibodies work is through the acronym PLAN. And this figure here again is just showing you that there are different types of antibodies. In this case, there are two antibodies, which notice they have very specific structures that are going to recognize very specific antigens. But how antibodies work is through this PLAN step of attack. So there's different ways in which antibodies can essentially work to kill whatever is invading our bodies and making us sick. So first off, we can have precipitation. Basically, what that means is that when our antibody physically binds to our antigen, that actually causes that whole situation, the antibody-antigen complex, to clump or precipitate together. So basically, they make this nasty big mass of pathogen, and that makes it really easy for our immune system to kind of perform a cleanup. So if you clump, it's kind of like putting all of your trash in the garbage can, and then the garbage truck comes and takes your trash away. Kind of the same idea. Another way in which antibodies work is through lysis or bursting. So we saw lysis when we talked about osmosis. Remember, this is when cells literally pop. Some antibodies can actually poke holes in the membrane of a target cell and kill it that way. So if you have holes, you've just disrupted the membrane. That's going to drastically affect what moves across that membrane, and that's going to kill whatever is infecting you. Some antibodies can attract phagocytes. Phagocytic cells are cells like the macrophage that are capable of engulfing large particles and chewing them up and getting rid of them. So macrophages perform phagocytosis, which is basically just cell eating. So these guys are they're gonna be attracted by this antibody, and they're gonna say, cool, whatever you're attached to, I'm eating it. And finally, some antibodies are capable of neutralizing the antigen. Sometimes those antigens are actually toxins, and if an antibody, and other antigens can be kind of poisonous or dangerous as well. So when the antibodies bind to them, they actually neutralize those toxins and prevent them from causing harm. This is often how bacteria work. So bacteria, we usually feel pretty crummy from a bacterial infection because they release toxins into our body. So antibodies can find those toxins and neutralize them. So again, in the antibody-mediated response, we have activated our B cells. Some of those B cells are going to become memory cells, which we're going to come back to, but the other cells become plasma cells. And it's those plasma cells which release antibody. So they release a ton of antibodies throughout the body. Those antibodies then travel through our blood, and they can attack their antigen through any one of these four options through the plan. They can precipitate, they can cause lysis, they can attract phagocytosis or phagocytes, or they can neutralize. So different antibodies are going to do one of these jobs. So that's the antibody-mediated response. Remember, the other response is that cell-mediated response, where again, after the alarm has been mounted, we have our helper T cell, and it's going to go on to stimulate a T cell this time. And specifically, it's going to get those cytotoxic T cells. Remember, or those are those killer T cells. So once activated, so remember, a T cell becomes activated into a killer T cell or a cytotoxic T cell. Just like we saw with the B cells, those T cells are going to divide. They divide into two populations. One of those populations will be the memory cells, which again, we're going to come back to. The other cells are going to be our effector cells. Remember, those are the cells that are going to go on and kill the pathogen. Remember that the cell-mediated response, this happens when a cell has been infected or it's abnormal. So in this case, rather than we are releasing antibodies into our system and having those antibodies go and find our pathogen, that entire killer T cell that we see here is going to go on and find whatever target cell has been infected. And when it does that, it basically attacks this cell that's being shown here. This is kind of the attack phase. But basically what is happening here is that in a simple sense, essentially punching a bunch of holes into the pathogenic cell. And that causes death of the cell. So these killer T cells, again, we have tons of them now floating through our bodies because we've divided, we've created 1,000, let's say. They're all going to kind of migrate through our body, look for these target cells, and then as soon as they find those targets, they're going to punch or perforate the target, and that's going to cause that cell to die. So in both cases, we are multiplying. We are creating two different divisions. We're creating kind of like a killer or a memory cell. If we're talking about those B cells, like the killer is those antibodies being released into our body. If we're talking about the T cells, we're actually multiplying and creating more killer T cells, and those are going to attack whatever target cell they're designed to attack. Now what I want to talk about in both cases, for both the B and the T cells, are those memory cells. And that's really step 7. So everything we've talked about up from step 1 to step 6 as a primary response. And again, the primary response is the first time that a pathogen is seen by our body. So I'm going to put seen in quotes because they're not using eyeballs, but they're detecting that target. And again, remember that when that primary response happens, there's only a few B or T cells that are capable of specifically recognizing and attacking that pathogen. But now we have just created, after that first response, remember that during that process, during that division process, we created memory cells. So now, after the first time, after that primary response, we now have kind of this collection of memory cells, and that's really important. So the primary response is usually slow, and that can be seen in this graph here at the bottom of my PowerPoint, where notice that you can have your first exposure to an antigen, say at day 0, and we see an immune response, and it's pretty big. That's a good immune response. We can see that we have antibodies being released into our bloodstream. And then notice that those antibodies do kind of decline over time. But notice that, they didn't peak. That antibody situation didn't peak until about, that looks like 25 days, right? So it took a while to get that immunity, which we're going to talk about what immunity is here in a second. So the primary response is slow. But after the primary response, because now with those memories, now we have a large supply of those memories, that's going to be able for us to react faster to future invasions of the same pathogen. And that faster second response is what is called the secondary response. And if we look at that portion of our graph, notice that the secondary response is almost like super, super fast. So in this case, notice that at day 60, this person was introduced to the same pathogen. And rather than there being kind of this nice slow arc to getting to maximum antibody concentration, it just went up. That looks like in 1 or 2 days, we have this huge concentration of antibodies. And notice that our second amount of antibody is much greater than the peak for the first. So we have considerably more antibodies in our system now. And that's really important because that allows us to very quickly attack and get rid of that pathogen, and we will never feel sick. During the primary response, you feel pretty crummy, which is actually a good thing. Having things like fever and the chills, as long as they're not prolonged, sore throat, runny nose, cough, all of those are actually symptoms of your body trying to get rid of what is making you sick. Obviously, a fever for too long can be really dangerous, so you don't want to have a very long-lasting fever. But feeling crummy is actually a good thing. That's also why with the COVID vaccine, they're saying that typically that second injection that you have to get, you might have fever and achy, kind of flu-like symptoms. That's good. It shows you that you have an immune system that's capable of doing its job. So it's okay to feel bad after a vaccine because it means your immune system's working. So we have primary responses pretty slow. We don't have as much antibody getting produced. The secondary response, because now we have a huge supply of memory B or T cells, we can mount a much faster response. We have way more antibodies being released and produced by our body, and that's going to allow us to get rid of that pathogen and not feel sick at all. And this is actually how vaccines work. A vaccine acts as the first or the primary response. But what's really nice about vaccines, which I've posted a video for you guys on D2L that just kind of is a quick 101 of vaccines on D2L. What's really nice about vaccines is you're not getting the actual pathogen that's making you sick. It's either just a portion of that pathogen or it's a dead pathogen. And that thing that is being injected into you is actually not going to make you sick. But what it does allow your body to do is mount a primary response. So that way, when you see the real thing, you already have memory cells that can then mount a secondary response and prevent you from getting sick. So vaccines are very good. Okay, so let's talk immunity. So immunity, essentially as a definition, just means you can be exposed to a pathogen and you don't get sick. And importantly, typically with immunity, that also means that the pathogen typically cannot survive in your body. So for example, most of us probably got chickenpox when we were kids. Chickenpox is caused by a virus, and chickenpox are miserable, right? You're itchy, you're just like covered in these hives, it's terrible. But while that kind of reaction is happening, your body has mounted a primary immune response. And afterwards, you have now those memory cells that live in your body, and you have immunity. And that means that chickenpox can't cause chickenpox in you ever again. And because of that too, because of that fast, quick secondary response, that virus is also probably being destroyed if you ever are infected by chickenpox a second time. So the pathogen can't survive in your body. That type of immunity, so when you have memory cells that are able to mount a secondary response in your body, that type of immunity is called active immunity. This is a long-lived thing. Some immunities last your entire life. Sometimes they don't. Sometimes you have to get like booster vaccines that kind of mount your memory cells and kind of bring them back up to speed. But typically, active immunity can last a lifetime for certain pathogens. And again, I posted a How Vaccines Work video on D2L just because with the coronavirus vaccine and with people having so many questions about it, I think it's worth just kind of posting how vaccines work. And I'm happy to answer any questions that you might have as well outside of class. So active immunity, you have memory cells, they can mount a secondary response. Passive immunity is a little bit different. Passive immunity occurs when a person receives antibodies that have been produced by another person. So for instance, during pregnancy, mothers pass their antibodies or memory cells onto the fetus via the amniotic fluid. So when a baby is born, they do have, while their immune system is kind of just getting started and up and running, because their mother has passed on parts of her immune system, babies do have at least some immunity. Babies can also receive antibodies and protection through breast milk, which is partially why breastfeeding is super important, especially in the first few months of a baby's life, because their immune system is not fully functional yet, it's just getting started. You get immediate protection from this, which is great, but it's very short-lived. It only lasts 3 to 5 weeks. So those antibodies basically essentially do degrade and need to be replaced. So while you have this almost immediate secondary response being able to be mounted in your body, it's not going to be for very long. And really, during pregnancy and breastfeeding is really the only times we see this passive immunity occurring. All right, so that is the immune system in a nutshell. The last thing that I want to talk with you guys about is partially one of the reasons why we jumped to chapter 13 so early in the semester. So you guys are going to be working on a final project really throughout the semester, and your first assignment is due this week. And really all you're doing this week is search throughout the semester and eventually present upon at the end of the semester. So what I've done, now that you have kind of an understanding of the immune system, I'd like you to look at the list of possible diseases that I've posted to D2L. And using that list and using the disease topic discussion in D2L, I'd like you to brainstorm with the class on what you think you'd like to present on. Everybody's going to present something different, but I want to use this discussion as a time to kind of get feedback from your peers of like, what do you think would be cool? Or what would you like to hear about? So make sure you're looking at the discussion and the list on D2L to get you started. I also want to point out that I am not covering chapter 13.8 in our lectures, but it does give some great ideas or examples of when our immune system doesn't function properly, and those could be potential diseases that you present on as well. And chapter 13.A, which is like a supplementary chapter to our immune system chapter, it starts on page 259. It also goes into discussion about different types of infectious diseases, so you can use that to help you as well. So use that list, use chapter 13.8 and 13.A to help you think about and brainstorm ideas. But that is it for this lecture. Reach out with questions and I will see you guys, I guess, virtually if you have questions.